Class, we studied about Mendel's first experiment, that is monohybrid cross. Today we are going to study about Mendel's dihybrid experiment.
Hello everyone. In the last video, we had learned the monohybrid cross of the pea plant and also the law of segregation, which said that out of the two genes, only one gene can be present in the gamete. In this video, we are going to see the dihybrid inheritance and the law of independent assortment. In dihybrid inheritance, it involves inheritance of two pairs of contrasting traits or two pairs of contrasting characteristics at the same time. The contrasting traits which were chosen by Mendel were shape and color of the seeds of the pea plant, and these were round and yellow and wrinkled and green seeds. Each and every seed of the pea plant would either be round or wrinkled, and it will also have a color either green or yellow. Both these traits will be present in each and every seed, which means that there will be no seed that will be missing these traits. It will surely have the combination of any of these two. Now for the dihybrid cross, Mendel took the purebred pea plant of round and yellow seeds and also a purebred pea plant of wrinkled green seeds. Again, we know that the purebred genotype has similar genes. So for the round and yellow, we have the genotype capital R and capital Y. Similarly, for the wrinkled green, the genotype will be small r and small y. When these two plants were crossed, Mendel observed that in the first generation, only round and yellow seeds were obtained and no wrinkled green seeds were obtained in F1 generation. Now we will use the Punnett square to understand how Mendel got this observation in the F1 generation. We know the genotype of both the plants. For this plant, the genotype is capital R and capital Y. And for this plant, the genotype is small r and small y. We know that the gametes formed will have one gene of shape trait and one gene of color trait. So the gametes formed for this plant will have genes capital R and capital Y, and the second gamete will have capital R and capital Y. Since two combinations are only possible here, therefore only two gametes are formed. Similarly, for this plant, two gametes will be formed which will have genes small r and small y, and the second gamete will also have small r and small y. So we have got the gametes. So let's draw the Punnett square to understand how we can fuse them. Since there are two gametes formed from each parent, there will be two rows and two columns in the Punnett square. Now I will write the gametes here. So it will be capital R and capital Y. Again capital R and capital Y. Similarly, I will write small r and small y. Again small r and small y. Now let's fuse them. So when I will fuse them, I will get capital R, capital Y and small y. All the cells will have the similar combination. So it will be capital R, capital Y, small y. Again capital R, capital Y, small y. And in the last cell also, it will be capital R, capital Y and small y. Now you can observe that the offsprings which are produced here have the similar combinations, which is capital R, capital Y and small y. And we know we have one dominant gene here which is of capital R and capital Y. So all the offsprings which were produced are round and yellow. Hence we can conclude that Mendel got all the round and yellow seeds in the F1 generation and no wrinkled green seeds were formed in the F1 generation. Then he took two plants from the F1 generation and crossed them. Unlike F1 generation, there were four different types of seeds with different combinations of shape and color in the second generation. These were round and yellow, round and green, wrinkled yellow and wrinkled green. Out of 556 plants, 315 plants were round and yellow, 108 plants were round and green, and 101 plants were wrinkled and yellow, and 32 plants were wrinkled and green. When he calculated the ratio of this, he got the ratio of 9:3:1. Now let's use the Punnett square to understand how Mendel got this ratio. Now we know the genotype of these progeny is capital R, small r, capital Y and small y. Now the gametes which will be formed will have one gene of the color trait and one gene of the shape trait. So let's form the genes here, gametes here. So the gametes will have gene 1 capital R, 1 capital Y, so it is the first gamete. The second gamete will have capital R and small y. The third gamete will have small r and capital Y, and the fourth gamete will have small r and small y. So if you look at any other combination, you will find that it will be just the repetition of these 4 gametes. So 4 gametes are formed. Similarly, for the second pairing also, these 4 gametes will be formed. Now we have got the 4 gametes, so let's draw the Punnett square, which will consist of 4 rows and 4 columns. So let's write the gametes here. So we will have capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. Similarly, on the left hand side also we will write the same gametes, which are capital R, capital Y, capital R, small y, small r, capital Y and small r and small y. Now let's fuse these gametes. So in the first, when we will fuse, we will get capital R, capital Y, again capital R, capital Y. In the second cell we will get capital R, capital Y and small y. In a similar way, when we fuse the third one, we will get capital R, small r, capital Y and capital Y. In the fourth row also, we will get capital R, small r, capital Y and small y. In a similar fashion, if we fuse all the other gametes, we are going to get these combinations. Now let's count the number of unique progenies which we will get here. So if we take in consideration the dominant trait of shape first, then we know only 2 types of progenies will be formed, which are round and yellow and round and green. In a similar way, if we talk of the recessive trait, then we know wrinkled yellow and wrinkled green progenies will be formed. So let's count them in the Punnett square. So first we are going to see the round and yellow one. Now we know for round and yellow, the genotype must consist 1 capital R and 1 capital Y. So let's count them here. So in the first row, you can see here, each cell has 1 capital R and 1 capital Y at least. So in the first row, we have 4 round and yellows. Now in the second row, you can see that the first and the third cell have 1 capital R and 1 capital Y. Similarly, in the third row, you can see that in the first cell and the second cell have capital R and capital Y. In the fourth row, you can see only the first cell have capital R and capital Y. Now, if I mark all the round and yellow in this Punnett square and count them, it comes out to be 9. Now, let's count the round and green one. For this, the genotype must contain at least 1 capital R and both the small y. So let's see in the Punnett square. In the first row, we don't have any such combination. In the second row, the second and the fourth cell have such combinations. In the third row, we don't have any such. And in the fourth row, you can see in the second cell, we have 1 capital R and both the small y. So if I mark them with the blue color, you will find out that there are 3 round and green. Now, when we count of the wrinkled and yellow, we know that the genotype must consist of at least 2 small r's and at least 1 capital Y. Now let's count them. In the first and the second row, you can see that there are no any such combinations. In the third row, you will find that the third and the fourth cells have such combinations. And in the fourth row, the third cell have such combinations. If I mark them with green and count them, then we have 3 wrinkled and yellow seeds. Finally, when we see of the wrinkled and green seeds, you can see that there in the fourth row, only in the last cell, we have both the small r and both the small y. So there is only 1 wrinkled and green. So the ratio of the progenies which will be obtained here is 9:3:3:1. If we look at the observation which Mendel obtained, he also got the ratio of 9:3:3:1. basis of this observation, Mendel concluded though the two pair of characteristics combine in the F1 generation, but they behave separately in the subsequent generation. With this, Mendel formulated the second law of inheritance, which was law of independent assortment. It stated that inheritance of more than one pair of characteristics in a cross simultaneously, the factors which are responsible for each trait are distributed independently to the gametes. Now for this dihybrid cross, we have taken the seed shape and the color, and we have seen that they behaved independently. Neither the shape affected the color of the seed nor the vice versa happened. So in this video, we have seen the dihybrid cross of Mendel and the second law of inheritance, which was law of independent assortment.
So far we studied Mendel's monohybrid cross and dihybrid cross, as well as we studied the laws of heredity proposed by Mendel. Now let's study the chromosomes, gene and DNA. The human body is made up of millions of cells. The nucleus of each cell contains thread-like structures called chromatin fibers. These are chromatin fibers. During the time of cell division, these chromatin fibers condense and form the chromosome. Each chromosome duplicate and form exact identical copies like itself, that is called as sister chromatids. During the cell division, each chromosome duplicates itself and form identical copies like itself. It forms another one copy, and these are held by centromere. So these copies are called as sister chromatids. These chromosomes are packed with DNA that forms the genetic material. The short segment of DNA is called as gene, that is responsible for the inheritance of particular phenotypic character. Each gene is located at the specific position in the homologous chromosome is called locus. During cell division, the genetic information present in the genes are passed from one generation to other generation. Now let's study the structure of chromosome. This is the typical structure of chromosome. The chromosomes are thin, long and thread-like structures. As I said you already, the chromatin fibers condense to form chromosome during the process of cell division. Each chromosome duplicates itself and form the identical copies of itself, that is called as sister chromatids. So these two sister chromatids are held together by centromere. So this is the typical structure of chromosome. This is thin, long, thread-like structure consisting of two identical strands called as sister chromatids. These two sister chromatids meet at a particular point that is called as centromere. Each chromatid is made up of the spirally coiled structure. This is called as chromonema. These chromonema contains many small bead-like structures. The number of bead-like structures are present in the chromonema throughout their length. That bead-like structures are called as chromomeres. These chromonema made up of the DNA, RNA, chromosomal proteins such as histones, non-histones, metallic ions. These things, these DNA, RNA, chromosomal proteins, metallic ions provide structural support to the chromosomes. This chromosome is first discovered by Valdeyer in 1888. Now let's study the different regions of chromosome. The first is the primary constriction. This is the primary constriction region. The two arms of a chromosome meet at a particular point. This is called as primary constriction or centromere. This is the region in which the spindle fibers get attached to the chromosome during the time of cell division. We know spindle fibers are formed at the metaphase stage and held the chromosomes and align the chromosomes on the equatorial plate during the metaphase stage. So that spindle fibers get attached to chromosome at the centromere region during cell division. So this is the primary constriction or centromere. Next is the secondary constriction. In some chromosomes possess many secondary constrictions at any point of the chromosome. This is known as the secondary construction, it is also known as nuclear zone or nucleolar organizer. That means in this is the secondary construction region. This is responsible for the formation of nucleolus of the nucleus. We know there's another spherical structure is present inside the nucleus, that is called as nucleolus. The next is telomere. The end of the chromosome is called as telomere. Each extreme end of the chromosome has a strong polarity. This polarity prevents the chromosomes from joining with the adjacent chromosome. This telomere maintains the stability of the chromosomes also. The next is satellite. So this is satellite. Some of the chromosomes have a knob-like appendages at one end. This is called a satellite. The chromosome with satellites are called as sat chromosomes. This is types of chromosome based on position of centromere. Based on the position of centromere, the chromosomes are classified as telocentric, acrocentric, submetacentric and metacentric. In telocentric, the centromere is present at the very end of the chromosome. They are rod-shaped chromosome. So in the telocentric, the centromere is present at the end, extreme end. It is rod-shaped chromosome. The next is acrocentric. Here the centromere lies near the end, near one end, and have a very small arm and long arm. Here the centromere, beyond the centromere, very small arm is there and one long arm is there. So this also rod-shaped chromosome. This is acrocentric. Next is submetacentric. Here the centromere lies near the center, not exact center. The centromere is placed near the center. Here also the two arms are unequal. Here one short arm and one long arm is there. It is J-shaped chromosome or L-shaped chromosome. The next one is metacentric. Here the centromere is present exactly at the middle. Here the two arms, both the arms are equal in length. It is V-shaped chromosome. The next is the classification of chromosome based on its function. In eukaryotic organisms, the chromosomes are classified into two types. They are autosomes and allosomes. The number of chromosomes in any living organism, either plant or animal, it is constant. It is species specific. Particular species contains the specific number of chromosomes. In human beings, there are 23 pairs of chromosomes or 46 number of chromosomes present in the cell. Out of the 23 pairs of chromosomes, the first 22 pairs of chromosomes are called as autosomes. These autosomes contain genes that determine the body characters. In sexually reproducing organisms, the autosomes occurs in pairs. Each autosomes occurs in pairs, and these pairs are called as homologous pairs of chromosomes. In these pairs, one chromosome is received from father and the other chromosome is received from the mother. Since these chromosomes occurs in pairs, this condition is called as diploid, and this diploid condition is represented by 2n. The last pair of chromosome is called as sex chromosome or allosomes. These sex chromosomes are determining the sex of an individual. There are two types of sex chromosomes. This is in females. The last pair is X and X. Two X chromosomes are there in the 23rd pair. But in the case of males, the 23rd pair contains the X and Y. Gametes produced by the organisms contain single set of chromosomes. That means the male gamete is sperm. The sperm contains 23 chromosomes, not in pairs, 23 chromosomes. That means the 22 autosomes are there. Along with the 22 autosomes, only one sex chromosome is there. So, I said you already, in males, the 23rd pair is X and Y chromosome. But in the gamete, along with the 22 autosomes, the sex chromosome either X or Y chromosome is present in the sperm. In some sperms have X chromosome 22A plus X, and in some chromosomes have 22A plus Y. This condition is called as haploid condition. It is represented by N. Like that, in the case of females, along with 22 pairs of autosomes, only one X chromosome is there. Normally, in females, in the body cells, the 22 autosomes and the 23rd pair may contain X and X chromosome in female and X and Y chromosome in male. This is the body cells. But in the reproductive cell, that means the sperm and the ova, in sperms, along with the 20 autosomes, either X or Y chromosome is there. Like that, in the case of female, along with the 22 homologous chromosome, any one of the X chromosome is there. So this condition is called as haploid. It is represented by N. Karyotyping. It is a technique by which the complete set of chromosomes is separated out from the cell and then they are lined up in an ideogram. Ideogram is the diagrammatic representation of chromosomes. This is the ideogram. Here all the chromosomes are taken out, separated out from the cell and lined up based on its decreasing length, thickness, position of centromere and shape, etc. This karyotyping is used to examine genetic problems as a case of disorder or disease. Home students are asked to study the following questions and write a home test. Answer in brief. First question, what are allosomes? Second, define heredity. Then you have to draw some diagrams in the classwork in a neat manner. The first diagram is the structure of chromosome, that is figure number 18.3 given in page number 265. Then a second diagram is the types of chromosome based on the position of centromere, that is figure number 18.4 given in textbook page number 266. Long answers. These long answers, students, you must study. No need to write this test. First question, explain with an example the inheritance of dihybrid cross. How is it different from monohybrid cross? Second question, explain the structure of a chromosome.